Given the last talk for this symposium is Mark Johnston. Dr. Johnston is a Professor Emeritus in the Department of Biochemistry and Molecular Genetics at the University of Colorado School of Medicine, where he served as chair from 2009 to 2019. He received his PhD from the University of California, Berkeley, after which he completed a postdoctoral fellowship at Stanford University. Dr. Johnston joined the faculty of the Department of Genetics at WashU, where he served as interim chair from 2002 to 2006. He was president of the Genetics Society of America and editor-in-chief of Genetics, and also is a member of the American Academy of Arts and Sciences and the U.S. National Academy of Sciences. Dr. Johnston's lab focused on understanding glucose sensing and signaling and developing methods for functional genomics, particularly for the analysis of transcription factor binding in the genome. His lab also played key roles in sequencing the yeast genome and the construction of the yeast gene knockout collection. Please welcome Dr. Johnston.
Okay, thank you. My talk today really has nothing to do about Gary. That's because I didn't know any computational biology, and all the other speakers before me have amply praised Gary's accomplishments and his contributions to our field, so I won't do that. I'll just preface my remarks by saying that I know that Gary was well known from the very beginning of the field. I was a graduate student in 1978. We were doing some sequencing, and we had a little bit of sequence, 100 bases or something. And we didn't know how to analyze it. And I remember somebody telling me, I can't remember who, but I clearly remember them saying, oh, you should talk to this guy Stormo in Colorado. He knows how to analyze sequence. I never found his email address, so I wasn't able to get in touch with him. What I want to do today, what I like to do today is to give you a history of why Washington University, and in particular the genetics department here, was really the founder of the field of genomics. So my talk is genomics instantiated. Now, that word instantiated, academics love that word. I've seen it all over the place. I've never used it, and I'm an academic, and I've always felt bad that I've never had an opportunity to use the word instantiated. So I want to thank the organizers for inviting me to give me the chance to actually use this word for the very first time. Now, of course, I had to look it up because I don't really know what it means. And don't tell me here. This will work at some point. Huh? I can do that, but I can't move it forward. Oh, I got to do this one. Okay. Yeah. Here we go. So I had to look it up, and I looked it up in the dictionary. Instantiate, to represent an abstraction by a concrete instance. And what I'm going to present to you today is my contention that the first instantiation of genomics happened right here up on the 8th floor of the McDonald Sciences building. And I'm going to tell you the story of how that came to be and how WashU came to be known as a DNA sequencing power. Okay, and I view from the front row. I had the privilege to observe all of this. I saw the whole story from the very front row. So I'll tell you my view of how it all happened. So back then, this is the 80s we're talking about, the goal was to map genomes. And so you have your genome here, and the goal is to get a single nucleotide resolution map of the genome. Well, back in the 80s, of course, you couldn't do that directly. You really can't quite even do it now directly. What you need is to map the genome first before you get it down to single nucleotide resolution. You have to cut the genome up into smaller pieces, figure out where they lie in the genome, and then you can get your single nucleotide resolution map. And it was those methods, it was that need to map the genome and how to do it that was really instantiated here in the Department of Genetics by this guy, Maynard Olson, faculty member who arrived here, I think, in 1978. And Maynard was really the first one to do genomics, the first one to study an organism on the whole genome scale. And what Maynard did was to map clones of the yeast genome. And he was the first one, along with really Johnston, who you'll hear about in just a minute here, to lay out the need plus the mechanism, the approach for how to map clones to genomes. And what Maynard did was simply do a restriction site digest of a large number of clones and measure the fragment sizes. So here's a bunch of clones of the yeast genome cut with EcoR1 and HindE3, run out on a gel to size the fragments, and you just count the sizes of the fragments. And if you do that enough times to enough clones, you can identify clones that overlap, that they share fragments of the same lengths. And so those clones must overlap. So if you're clever enough to be able to run gels that can accurately size DNA fragments, and then you're clever enough to write algorithms that can handle the data and put it all together, you end up with something that looks like this, a clone map of the genome, where the horizontal lines are individual clones. The hash marks on the clones are EcoR1 and HindE3 sites. And so you can generate a series of clones that physically map the genome. And Maynard was the first guy to do this. And he was doing it at a time when nobody could understand why you'd want to do that. I remember a very well-known scientist, very smart guy, who said to me, who the hell cares where all the R1 and HindE3 sites are in the yeast genome? I don't really care. Well, the reason you care is because you need a map of a genome to get a sequence of a genome and then all that follows from that. Okay, so Maynard was the first one to map genomes. Maynard was working at this level in the range of clones of about tens of kbs in phage vectors and plasmid vectors, and later on in cosmid vectors. He wasn't working at this larger scale of hundreds to thousands of kilobases of DNA. So now comes the key event that really led to WashU having a genome sequencing center. So you all know that it's much easier to put together a puzzle of a few number of pieces than a puzzle with lots of pieces, right? A 1,000-piece puzzle is much harder to put together than a 100-piece puzzle. And Maynard was working in this range. And then comes the key event. Bob Waterston, Maynard's colleague and my colleague up on the 8th floor of McDonald Sciences building, goes off in the mid-'80s, 85, I think, 86, to do a sabbatical with Johnston. And Bob's over there working on his muscle genes in Bob's and John's lab, watching John map the C. elegans genome. And so John was doing the same thing Maynard was doing, trying to get a map of the C. elegans genome eventually for the purpose of sequencing it. And John was also mired in this area of small fragments. And he had 750 contigs. There were 750 gaps in the clone map when he should have had 7, right? And he just couldn't get past those 750 gaps in the DNA sequence. And Bob is watching John be frustrated with that. Bob comes back to St. Louis and he talks to these two guys, David Burke and George Carl. They were graduate students in Maynard's lab. And they told Bob about work they were doing to generate yeast artificial chromosomes. These were vectors that would take large pieces of DNA. And Bob learned about this before it was published. Bob goes back to John Sulston's lab in Cambridge, England, and begins to work with John on closing those 750 gaps in the genome sequence. And that's really what got Bob into DNA sequencing, which is what brought a genome sequencing center with hundreds of millions of dollars in grant income to Washington University. Okay, so there's another landmark. So we have clone mapping from Maynard and John Johnston. We have yeast artificial chromosomes from David Burke and George Carl, again, working in Maynard's lab. And there's a third major landmark in genome mapping that also came out of Maynard's lab called sequence tagged sites. And so Maynard in 1989 publishes this paper with a few other people to give it credence, I guess. A common language for physical mapping of the human genome, sequence tag sites. And sequence tag sites are simply PCR priming sites that will yield a PCR product. So you simply get random sequence from a bunch of clones, design primers, and if those primers amplify, give a product, amplify a sequence, then that means that clone that was the template has those sequences in it. So it's simply sequence landmarks of about 100 to 1,000 base pairs. Sequence tag sites, really a brilliant idea, quite simple, which is as most brilliant ideas are. They're brilliant after you know what the idea is. And the guy who put that into practice, sequence tag sites into practice, who instantiated sequence tag sites, was this guy, Eric Green, a postdoc with Maynard. Eric was a student here, an MD PhD student, got his degree in immunology, and decided he wanted to go into genomics and join Maynard's lab and really put into practice Maynard's idea of sequence tag sites, and ended up generating by 1990 a map of a region of the human genome. It was a model for how you map the human genome with sequence tag sites. So here's all the clones, the horizontal lines, and now those dots on the clones are sequence tag sites, PCR priming sites that are present in those clones. So you generate a map of the genome that way. Eric went on to become the second director of the National Human Genome Research Institute, where he has been the director now for 15 years. So genomics was born in the genetics department, was extended by the sequencing center in the genetics department, and then we exported it to the National Human Genome Research Institute. Mr. Mack would have been proud that his money invested in the genetics department really came to fruition. Okay, so now we've got a map, and now we're ready to do the sequencing. And you all know how this works, right? Dideoxy nucleotides get incorporated, cause chain termination. You generate a family of molecules that have each stopped at a nucleotide, and you separate those by their size on a gel, and you read the sequence off. Now, this was how it started for C. elegans sequencing. I remember them developing films and reading off the sequence. I remember, you know, fishing gels out of the fixer and reading the sequence before they were even dry, because you couldn't wait to see your exciting sequence come off. Now, of course, you're not going to be able to sequence a genome that way, right? You got to have some automated way to do it. And so a great leap forward came with the development of fluorescent tag DNA for sequencing, where you put on your dideoxys some dyes that report a color, and now you can load all of those reactions into one lane and simply read off the sequence from the color of each of those bands. So that was a major advance that allowed automation and led and sort of portended that the sequencing of the human genome was going to be possible. Get these kinds of gels where you read off the sequence. And here's really what the readout was, right? These peaks of these colored peaks are the peaks of those bands coming off the gel. And you just simply read the sequence off from the color. But the problem is, where do you start your sequence reading? Down here, it's not going to be a very good sequence. It gets pretty good in here, but then the sequence starts degrading. Where do you stop reading your sequence. And here's where a major advance came that really enabled the human genome sequencing. And it was also comes from the Department of Genetics on the 8th floor of the McDonald Sciences Building. And that is the development of quality statistics by Phil Green, who was a faculty member in genetics. Here's Phil. And what Phil did is develop algorithms that would read those peaks, those colored peaks, and put a quality score on them. So his algorithm could read the peaks and tell you how sure of each base call you are. So Phil put a score on each of those peaks. So a score of 10 means that there was a 1 in 10 probability the base was called wrong. So you're 90% certain that that base is called right. A score of 50 said that there was a 1 in 100,000 chance that you were wrong. So you're almost certain of that base call. And so that enabled you to turn these peaks into scores, which enabled automation of the sequence reading. And that was a major advance in the sequencing, fluorescent sequencing of the human genome, a major advance. There were other advances. This is the slide I got from Maynard. These are Maynard's landmarks in the evolution of 4-color sequencing. There were others, advances in polyacrylamide gel technology, better dyes that were more, gave you more sensitive detection, DNA polymerases that would accept better the dyes, the dye-labeled nucleotides. And there's one more advance that was critical that, again, also came from Washington University. This one not from the McDonald Sciences Building, but from the first floor of the North Building in the biochemistry department. And that was the development of cycle sequencing, using PCR technology to amplify your sequencing reaction so you can sequence small amounts of DNA. And that was done by our late colleague, John Majors, and his student, Jeff Axelrod. And so here's John. And what Jeff and John did was they used multiple rounds of annealing and extension with Pac polymerase to detect sequences in the genome. So you do a one-sided PCR reaction, essentially, a linear amplification of the reaction. And John pointed out that their primary extension assay could be used to determine DNA sequence directly from the yeast genome. And so that was the first instantiation of cycle sequencing, which was another major advance that enabled the sequencing of the human genome. Okay, so mapping a genome happened here. Many other advances happened here at Washington University. So why am I up here telling you this story? It's all about Maynard and Bob and Phil, right? Why am I here? Well, I had a front row seat because I was peripherally involved at the project. So when Bob set out to sequence the worm genome, he and John decided they needed to get the sequence of the yeast genome also. Why? Well, they thought that they were going to be sequencing yaks, C. elegans DNA cloned in yaks. And if you isolate yak DNA from yeast, you're going to necessarily be contaminated with yeast DNA. So you got to know the yeast sequence so you can throw it out. So they wanted to sequence the yeast genome, and so they recruited me to be in charge of that project. And so this was our first paper on this, 1994, sequenced one of the yeast chromosomes. This was the second whole chromosome sequenced. And that's why it was published in a relatively high, in a ostensibly high profile journal. And it was done, we did it in quite quick order because we had Rick Wilson and Bob Watterson Sequencing Center that enabled us to do that. Now, what does it mean to manage a project like that? So my role to manage this project was to rely on Linda Riles. Linda was Maynard's technician who played a big role in his mapping of the yeast genome. So Linda was the caretaker of all the clones of the yeast genome. Here's Linda in front of the maps of the yeast genome. So my role was to go tell Linda what chromosome we were going to sequence next. Linda would go in and figure out what clones spanned the chromosome. She'd give them to me. I'd walk them the 3 blocks over to the 4444 building, hand them to Lucinda, and they'd get sequenced. The sequence would come in, and I had the privilege of sitting in front of a computer and annotating it, which was really fantastic. And I got all the credit. It's great work if you can get it. Highly recommend it. And then just a little while later, we collaborated with a worldwide consortium of sequencers. Back then, sequencing was hard, and so lots of people were involved to produce the sequence of the first eukaryotic genome, yeast, about 12 megabases in size. This was an interesting sociological and political science experiment also, which we can talk about later maybe. Then soon thereafter, Bob and John's sequencing center sequenced a real genome, a large genome, about 100 megabases, about roughly 10 times the size of yeast. The first multicellular organisms whose genome was sequenced. And then, of course, you know that they sequenced the human genome. Here they are in 2000 celebrating the completion of the yeast genome. Here's Bob Watterson and Rick Wilson at the White House with a few other minor players in the process, celebrating what they called the completion of the yeast genome, of the human genome sequence. So really, this guy was really, I think, one of the moral compasses of the project for data release, for appropriate allocation of the resources of the project. He really, I think, does not get all of the credit that he deserves for his leadership role, his quiet leadership role in that project. Here, this is Bob at a press conference with Craig Venter talking about the sequence. So that's how genomics was instantiated in the field. Genomics was invented, I claim, here at Washington University on the 8th floor of the McDonald Sciences Department by Maynard, implemented extraordinarily well by Bob, and advanced by people like Phil Green. So we should all be proud to have been part of Wash University. And I had the great privilege of having a front row seat for all of that, which I've just told you about. Thanks.